All right, guys. So I am back at the classroom today. So first video I'm filming, back at Albert Gallatin. So what I'm going to do today is we're going to go over some more structures inside the cell. But before we go over that, this room echoes so much more than the one in my house. It's crazy. But anyway, before we go over that, what I wanted to do was go over surface area to volume. I'm not going to do another example. You guys know now, as our volume of the cell increases, our surface area doesn't increase proportionally, which gives us a lower number for our surface area to volume ratio, which we know that's not good for the cell. So we want to keep the cell fairly small. The other thing I wanted to show you guys is this is your equation sheet that you will get when you take the AP Bio exam in May. Now if we look down here, all the way at the bottom, we get our surface area, that kind of thing, of a rectangular solid. We used a cube. Okay, cube's down there. But anyway, so here is your surface area of a sphere. I just want to make sure you guys saw that was on there. So in case they do give you a sphere, you just put it into this equation. Obviously, they'll probably give you the radius of the sphere. So you would just put the radius in there, square it, and then you can use pi or not use pi. It doesn't really matter because you're going to be doing the surface area to volume ratio, so pi would eliminate itself. But anyway, I just wanted to show you guys that. And you can Google this. You could just go to Google and say AP Bio formula sheet, and you can look over that. Okay. So last time we left off going over the pathway of a protein, and we started with the nucleus. Then we went to the rough ER, then we packaged our contents up into a vesicle, then that vesicle went to the Golgi and fused to the Golgi, actually became later part of the Golgi. Our protein went through the Golgi, and then lastly, once it was done with the Golgi, it left the cell through the cell membrane. So now we're going to go over the lysosome. What the lysosome is it's pretty much like the recycling center, we could say, of the cell. So it has a lot of enzymes in it. And what these enzymes can do, guys, is they can break apart maybe whole cells, maybe just part of a cell, okay? Like maybe a macromolecule, something like that. But what's nice about this is breaking apart our proteins, our fats, the polysaccharides, and the nucleic acids, as it says there. What's nice is if we break them apart, then we can use those parts, that contents, those elements, to make other macromolecules later on. The lysosome, it's slightly acidic, not like really strongly acidic, but slightly. It's about a 4.5 to a 5 on the pH scale. So nothing too crazy there, but that's best for those enzymes. If the pH gets too high, maybe gets to 7 or becomes maybe even basic, those lysosome enzymes are going to become denatured and they are not going to function properly, or maybe they won't function at all. So I got a little video on phagocytosis. We'll go over that video in a minute. But what can happen here, guys, is cells can engulf other cells. Well, why would we want to engulf other cells? It seems weird. It's like we're eating our own cells. Well, maybe something's wrong with that cell, all right? In this example, we have WBCs here, which are white blood cells. You guys know white blood cells, they play a role in immunity and fighting off infections, fighting off bad things in your body. So why we would use the white blood cells here is, okay, we got a bad cell. Maybe it's infected with some disease or something. Maybe it has little viruses inside of it that we don't want those viruses coming out of that cell. So we're going to go through phagocytosis, and we are going to get rid of it. Okay. Other thing, guys, with the lysosomes, just something to point out here. There's a disease called Tay-Sachs disease. It's a genetic condition. People are actually missing the enzymes in. Hold on.
Mr. Pegg, if you're in the building, please call Tina at 5301. Mr. Pegg, 5301.
It's a problem with being back at school. We have announcements now. I have interruptions. But anyway, so Tay-Sachs disease, like I said, genetic condition. People are missing the enzymes in their lysosomes. It's a specific enzyme. It's one that's going to break down the fatty substances, the fatty acids, which, yeah, that's not good, okay? We want to be able to break down our fat. Problem with this is, if we don't break down the fat, we're going to get this fat accumulation in mostly the brain, which is, yeah, not bad. It affects the nerve cells, and people with this condition, guys, they usually die within like 3 to 5 age. They have a very low life expectancy, so kind of sad. So here is our animation of phagocytosis. You guys can see something is coming in. Okay, they have like a little food vacuole here full of food. And what happens here is our lysosome is able to take that food and it's able to break it down. So a little bit of a different example than the white blood cell one. The white blood cell one is trying to kill it in order to make sure, hey, infection or whatever, the bad bacteria doesn't spread through the rest of your body. And here's another one, guys, of a peroxisome. We will talk about what a peroxisome is later on. There's a specific slide on it. But you guys can see once again that the enzymes inside the lysosome are breaking the peroxisome into smaller parts. So here's a little video on phagocytosis, just so you guys can see the animation behind it.
Welcome to another Patho video. Today's topic is phagocytosis. Phagocytosis allows phagocytic cells, such as macrophages and neutrophils, to ingest and kill invading pathogens. The process of phagocytosis follows 3 main steps. First, recognition and adherence. Second, engulfment.
Can I have your attention,
Jeez, my goodness, sorry guys.
staff, teachers, employees, anyone that is interested in a flu shot? We have someone from John Eagle here in the cafeteria that would be happy to give you
Flu
your
shots.
shot. Thank you.
I apologize, guys. This is crazy. They were offering teachers flu shots. Lovely. I don't believe in flu shots.
And finally, intracellular killing. Direct recognition and adherence occur when a pathogen-associated molecular pattern, or PAMP, expressed by a microbe, binds with a toll-like receptor on the membrane of a phagocyte. During engulfment, extensions of the cytoplasm move around and eventually enclose the particle in a membrane-surrounded phagocytic vesicle, or phagosome. Once inside the cell, the phagosome fuses with the cytoplasmic lysosome, forming a phagolysosome. The lysosome contains antibacterial molecules and enzymes that can kill and digest the microbe. Intracellular killing is accomplished through several mechanisms, including toxic reactive oxygen species, such as hydrogen peroxide, and nitrogen-containing species, like nitric oxide. Any indigestible material is then removed through exocytosis. Here's an animated summary now, showing the steps of phagocytosis. This concludes the video on phagocytosis. Thanks for watching.
All right, guys. So, not the most energetic person in the world, but his animations are really good. Okay, so that's it with phagocytosis, guys. It does involve the lysosomes because we're bringing things in the cell through the cell membrane, and then the lysosome may break them apart into smaller pieces. Moving forward to the vacuole. So the vacuole are going to be large vesicles. They're no longer going to be used to transport proteins, but they kind of have like the same shape, especially in our cells. Plant cells are a little bit different. But anyway, So we're going to have some different vacuoles we're going to talk about. Let's talk about the plant one actually first, which is the central vacuole. The central vacuole in plants, it's going to take up most of the cell. If I wanted to draw it for you guys here, if we have a plant cell, I will make it green here. Here's our plant cell. We are going to have a central vacuole, which usually isn't in the center, but it's going to be gigantic. There's your central vacuole. Main thing, guys, is it's going to be able to contain a lot of water, so that way if the plant doesn't get watered every day, or you forget to water it, or there's a drought outside, it's going to have a nice store of water for whenever it needs it. The other one here, guys, is the contractile vacuole. So here's what is going on with the contractile vacuole. We have some kind of amoeba, protist, some kind of organism, and it's living in water. So I will just draw a glass of water here. All right, here's my lovely glass. Yeah. You do real good drawings, Yeah, they're horrible. Sorry, guys. Okay. So here is our little protist or amoeba, whatever. Inside of it, and I will draw it in green, we're going to have this very tiny contractile vacuole. And just like the word states, guys, it's going to contract. Now, what's going to happen here, guys, is this cell is living in a very odd environment. And we'll go over the terms hypo and hyper later on. But you guys know solute and solvent. So we have some solute particles in the water here. In our amoeba, our protist, we are going to have a lot of solute particles. Okay? What happens here, guys, is water. Oops, sorry, the lights went out. Give me a second here. I will tell you, it is so much harder to film a video here. Anyway, so what's going to happen here, guys, is water is going to move where there is more solute. So water is going to move inside of the cell. That's going to make the cell get bigger. Problem is, amoebas, protists, organisms like this, they're only made of one cell to begin with. So if that cell gets too big, it's going to burst and the organism's going to die. So what the contractile vacuole does is throughout the whole organism's lifetime, it's going to get real big, fill with water, and then spray all the water outward. And then it's going to keep repeating this process over and over and over again, where it fills with water and ejects the water from the cell. That way it does not explode. So that's the contractile vacuole. All right. There's a good picture of a central vacuole, guys, in plants. All right. We'll go over a little bit of the endomembrane system, and then we'll call it a day. So, well, I'm sorry, guys. We already went over that. Endomembrane system, that's the one where we made all of our proteins and everything like that. So we're going to go and do the different organelles in the cell that help us to generate energy. So the two main ones are the mitochondria and the chloroplasts. We'll get into peroxisomes later on. The main goal of the mitochondria is to produce ATP. And if you remember from the last chapter, the ATP, we have the 3 phosphates there. We're going to break a phosphate off, and that releases energy and transfers the ATP into ADP. Chloroplasts, which are going to be plants in plant cells and in algae, but not in our cells, this is going to be the site of where our plants, where our algae is able to make glucose, able to make sugar. And then the sugar is then going to be used by the mitochondria to convert it into ATP. Okay. So similarities between the mitochondria and the chloroplast, both have a double membrane. They both have ribosomes and a circular DNA molecule. and they both grow and reproduce independent of the cell. So because of this, sorry guys, I thought there was going to be another announcement. Oh my goodness.
The following teachers, please report to the cafeteria for a brief meeting. Mrs. Addis, Mrs. Brooks, Mr. Colbank, Mrs. Communell, Mr. Karofsky, and Mrs. Mallon. Please report to the cafeteria. Thank you.
We will get through this, guys. My goodness. Okay. So for this, we have the endosymbiotic or endosymbiont theory, however you want to say it. It's fine. But what this is saying is once we had a ancestor of a eukaryotic cell, and it engulfed a prokaryotic cell, and then they had a symbiotic relationship with one another. They helped each other out, okay? They think that the mitochondria and the chloroplasts were once, prokaryotic cells, okay? Now, given this, guys, we think that they were aerobic, okay? So there's, here, let me write them down here for you guys. There is aerobic, which is like oxygen. And then there's anaerobic, which is no oxygen. We think that the mitochondria and the chloroplasts were anaerobic bacteria, anaerobic prokaryotes. So we think they're this one, all right? And then they were taken in by the larger cell. They were taken in by the eukaryotic cell. Okay, so that's why we think, that's what we think happened based on, if we go to the previous slide, they have a double membrane. It kind of resembles the cell wall and the cell membrane in prokaryotic cells. They have a circular DNA molecule that's just like prokaryotic cells. They have one big circular DNA. And they grow and reproduce separately, so that still would be kind of prokaryotic. So that's the endosymbiotic theory. Here is a picture of that. Okay. Cool. All right. I'm going to draw some pictures for you guys, but I think I'm going to wait till tomorrow on that because I just keep getting interrupted and I'm not getting as far as I want to. So I'm going to try to film one later, guys. All right, have a good day.